Thank you. I'm pleased to share with you some recent results on studying retrograde tracer-based connectivity and comparing it to functional connectivity in the Macaque. And this is a collaboration with the labs of Takuya Hayashi and Henry Kennedy in my lab. So let's remind ourselves to start with that resting state functional connectivity is widely used to infer how strongly connected different brain regions are from many presentations at this meeting as well as a large literature. But we also know that functional connectivity is a quite indirect measure based on correlations of bold fMRI signals that lead to connectivity matrices that may be dense or parcellated as the analysis goes. But when one or another study or an investigator finds a difference in functional connectivity between groups or between conditions, the question is fundamentally, does that reflect genuine anatomical connectivity differences or something else? And to begin to address that more incisively, we need quantitative comparisons between functional connectivity and anatomical-based connectivity measures. That's not readily available in humans. We don't have any ground truth for long-distance connectivity data now, and we're not likely to get any anytime soon. We do have the alternative of tractography from diffusion imaging, which is in some ways arguably a more direct measure of wiring in the brain leading to connectivity, but as my lab and many others have shown, it has pretty significant limitations. So instead, what we've taken under our belt for the present analysis is to take the macaque monkey as our model system and use tracer-based connectivity from the Henry Kennedy's lab that Yuji Hu is presenting in a poster 698 this afternoon, and functional connectivity analyses from Takuya Hayashi's lab, poster 676. We started with baseline kind of ground truth information from the Markov et al. Study several years ago, where retrograde tracers were injected into 29 cortical areas, and for each of these areas there was a determination of the fraction of extrinsically labeled, retrogradely labeled neurons for each of the 91 source areas in the parcellation they used. This led to over 1,600 pathways, which is more than 60% of the possible number in the 29 by 91 connectivity matrix that was used. But importantly, there was literally a 100,000-fold range of connection strengths from the very strongest to the very weakest for each of these connectivity profiles. And in addition, the parcellation is a relatively coarse and not a complete ground truth accurate parcellation. So in approaching this next stage of our analysis, we wanted to use an unbiased parcellation-free approach. And the way we did this was to take densely spaced histological sections. On each section, the distribution of retrogradely labeled cells was marked one at a time. And we took the section contours, the peel and mid-thickness and white matter surface contours, and found the best matching corresponding slice and orientation from the surface-based atlas that my lab has generated, the Yerkes 19 atlas from MRI data. And we mapped the retrograde tracer density onto the MRI volume and then mapped into the individual surface and represented the results on the inflated maps that you see here. So this is an example of a ventromedial prefrontal cortex injection. You can see the distribution at a very fine-grained dense level. Here's another one quickly, a dorsal medial prefrontal cortex and a dorsolateral prefrontal cortex injection to see the fidelity of the representation that we can attain. And we've done this for 31 tracer injections to date and mapped the data onto a surface-based atlas. Now, if we jump over to resting state fMRI, we know that fMRI gives relatively weak signals that are buried under quite a lot of noise and bias and artifacts. In human analyses of fMRI data, what we call the HCP-style acquisition and analysis strategy is aimed at getting the best signals possible and minimizing noise and getting rid of biases using approaches that Matt Glasser's papers articulate. Working with Takuya Hayashi, we've extended this HCP-style approach for the macaque, And that involves a customized head coil for the macaque, resting state fMRI data. In this study, 102 minutes from each of 30 anesthetized macaques, and we worked to titrate the anesthesia to be relatively light but stable to get good signals. And then we carried out pre-processing that corrected for motion and distortion, mapped to Sifty-Gray ordinates, and then other pre-processing to do spatial ICA with the 26 component analysis and various state-of-the-art strategies to get high-quality functional connectivity. And then we mapped the data to the same Yerkes 19 surface-based atlas that you saw for the tracer-based analysis. So let's take a look at an example seed location in anterior cingulate cortex, and that reveals functional connectivity with a broad swath but a very detailed pattern in somatomotor cortex regions mainly. And if we move the seed location slightly anterior, then we see a dramatically different functional connectivity pattern indicating that we have preserved high spatial specificity in our mapping. But we now want to compare this to anatomical connectivity by putting our tracer and injection sites matched to the seed locations for functional connectivity. So on the left you see the results for a tracer injection in area 25, and you can see a high density of retrograde tracer label in various anterior regions and in posterior cingulate cortex. And lo and behold, on the right, you see a fairly good match that where there's high tracer density, we also get high functional connectivity. But there are some regions shown in green arrows where there's moderate functional connectivity and no tracer connectivity directly indicated. Here's another example where there's also a high correlation of 0.59, the previous one was 0.58. This is an area 7B injection, and again, where there's high tracer density, there's strong functional connectivity, but other regions that show moderate functional connectivity and no tracer labeling. Two examples quickly that show the opposite or much weaker correlation patterns. Here's an LIP injection on the left, and where there's strong tracer, there is strong functional connectivity, but there are much larger regions of moderate functional connectivity or even strong functional connectivity and no tracer labeling. And the last example has a very weak correlation of 0.06 from a prefrontal cortical injection that does have functional connectivity where there's tracer, but even more broad and more moderate functional connectivity in the absence of direct tracers. So one possible explanation for the broader distribution of functional connectivity is that it reflects indirect as well as direct connectivity. So for example, if this anterior temporal projection back to prefrontal area 25 receives inputs from more posterior regions, that might account for the functional connectivity in that region. A possible strategy for addressing that was proposed by Steve Smith, which is to use partial correlation. And we use the 26-dimensional ICA data to drive that analysis. I'm not going to try to explain it here. There's a brief description in the text that involves regressing out the 24 ICA components from each of the seed and target pairs, preserving the relationship within the directly the strongest correlated ICA components. But if you want to know more about that, please come to the poster. Let's just ask, does this strategy of partial correlation work? And the answer is yes and no. And what I mean by that is shown here. So for each of the 31 tracer injections that we have, the blue values on the histogram show the full correlation analysis, and it ranges from a high of 0.612 down to the low of 0.06 you saw before. For the partial correlation analysis shown in pink, there's some regions where the partial correlation analysis is actually not as good as for the full correlation, but there are other regions particularly in prefrontal injection sites where partial correlation outperforms full correlation. But overall, they're pretty much very similar, 0.42 versus 0.39 for the average correlation. And the last scientific point to be made is to look at the dependence on distance, length of the pathways as assayed using the tracer analysis to compare functional connectivity as a function of distance. And what you can see is that for full correlation analysis, when we do a parcellated analysis that Takuya Hayashi carried out, there's an overall correlation of 0.65, but for the long distance pathways in yellow colors, the correlation is still actually quite good, 0.61, which is not something that we saw when we compared tracers versus tractography. And lastly, for partial correlation analysis, there's again a reasonably strong correlation for full correlation as well as partial correlation at long distances. So to conclude, functional connectivity is generally high between strongly connected regions, but it's also positive in regions that, many regions that lack direct connectivity. The role of indirect second-order pathways versus common inputs that might include neuromodulatory sources is something that would be very important to determine, and we're hoping to provide this data set to make it publicly available to let others help investigate the best algorithms for estimating connectivity. So I'd like to thank the collaborative team, Henry Kennedy's lab, Yuji Ho presenting a poster here, and Takuya Hayashi and Yunus Atio and others in the Riken Kobe Institute. And I'd be happy to answer any questions.